Okay, so yeah, I'm going to be giving some information on the first phase, some results from the first phase of this research, and talking about the development of this second stage of developing a better monitoring tool for where mountain pine beetle is in low densities at this edge of expansion. So I'm going to go kind of fast through this background because I presented some of this last time. But during colonization, pheromones are needed for that mass attack behavior of mountain pine beetle. And because of that, we've been able to use these synthetic pheromones as monitoring tools. But in this situation in Alberta, where populations are low and it's in a novel habitat, we're unsure about the effectiveness of these pheromone formulations that we use. But along with the pheromones are also the host compounds, the volatile host compounds. So during the dispersal, host selection, and the aggregation and host colonization part of mountain pine beetle's life cycle, these volatile chemicals from both the trees, hosts, and from the beetles are needed for these behaviors to happen. And there's three features of bark beetles' relationship with their hosts that are influenced by these secondary host compounds. The first one is that beetles are required to kill the tree. They need to overcome these defenses to successfully reproduce. The second part of this relationship is that some bark beetles use these host compounds to locate hosts. And the third important part is that this pheromone communication is closely linked to these host compounds, and this is mostly monoterpenes, where monoterpenes are acting as the precursors for mountain pine beetle pheromone, or as a stimulant for a mountain pine beetle to make these pheromones. So together with these monoterpenes being the basis for pheromone production, and also that they synergize bark beetle pheromones, they're an important component that we need to look at while they're in this novel habitat. So to quickly go over just the attack sequence and why pheromones are and host volatiles are important. So females are the initial attacking beetles for mountain pine beetle. They start producing transverbenol, which is the aggregation pheromone, and together with host terpenes, which synergize the effect of transverbenol, they attract conspecific, so females and males. And then during the attack, when there's both females and males there, the males start producing exobrevicomine, and together all of this component attracts a lot of beetles, and this is that mass attack behavior that we see. But at some point, the tree fills up, and anti-aggregation pheromones are made, and that directs beetles to attack adjacent or neighboring trees. But we're interested in these aggregation pheromones and how they can be used in management. So trap trees is what I'm going to mostly focus on this talk with. So trap trees, the idea is that you can bait it with mountain pine beetle pheromones, and beetles are attracted to it and attack that tree, and then before the beetles the next year beetles come out, the brood come out, you remove that tree, thereby essentially removing those brood beetles from the population, from the local population. And so these trap trees can be used for detection of presence and monitoring of population fluctuations, but also we can use them as part of a more direct control strategy where we can use trap trees to concentrate beetles into baited areas and also prevent a spread from baited areas. And this has been used in BC, it is used in Alberta, and it's also been successfully used in other beetle species, southern pine beetle and Douglas fir beetle too. However, there's some issues in that the distance to an active beetle population is a factor in whether you can get this to happen, and then with these selected stands, cut them and remove that population from being able to attack adjacent stands, and also the formulation of what you're using to attract the beetles to that to those trap trees is also an unknown for us, again, in this situation with low populations of mountain pine beetle and in a novel situation, novel habitat. In Alberta, as I said, they're using these trap trees, especially in this leading edge zone, and they use it as for detection. And they use 3 trap trees spaced 45 meters apart in a triangle, and they have these placed throughout here. And but again, it's not really known whether this is the most efficient configuration of trap trees. Is it enough trap trees? And can these trap trees be used to concentrate beetles and prevent their spread, which means they could be used for a direct control strategy. And so that's where this project comes in. So I'll be presenting some results on this phase 1, where we looked at the formulation of these aggregation pheromones and tree volatiles to attract beetles in novel habitats. And what we learned in phase 1 will apply to phase 2, which will start this summer and be repeated in 2016. And this phase 2 is to develop a field protocol for deployment of these lures on trap trees to monitor and detect mountain pine beetle populations, but also to evaluate the use of these trap tree protocols to look for indicators of annual trends for population fluctuations, and also as part of a direct control strategy, in that do these concentrate beetle populations and prevent them from spreading to adjacent stands. So for this first part, we put out some trap lines, and on that we used the commercially available mountain pine beetle pheromones, so that's transverbenol and exobravicomin, and we varied the tree component, the tree volatile component. And we did this study this last summer in the Swan Hills area, and we had 2 experiments. One where we used the commercially available mountain pine beetle pheromone plus 1 of these volatiles, and the second one was the second experiment was where we used the commercially available pheromone plus terpinolene, and then one of these compounds. The terpinolene plus the pheromones is generally what is used for lures for these standing traps. So in this graph, I have the different treatments with the bait, which again is just transverbenol and exobravicomin, plus 1 of the tree compounds. And we found that the addition of myrcene performed just as well as the addition of terpinolene, which again is the standard lure. But part of this project was to look at the percentage of females caught, because the greater the percentage of females caught, the more you're able to affect the population. And we did see a little bit lower percentage female catch with myrcene. And then for the second experiment, which is the standard lure plus terpinolene and then one of the tree volatiles, we found that the standard lure plus myrcene performed better than just terpinolene alone. And also 3-carine had some promise, and also myrtenol. And then when looking at the percentage females caught, even though the myrcene treatment here caught a lower percentage of females than the terpinolene alone, the number of females caught with this treatment was still greater. So not only can myrcene be used in place of terpinolene, but also terpinolene and myrcene together actually caught more beetles. And this has also been shown in some recent research. So this is this data supports some other research. So that is our best performing lure that we are going to use this summer and next summer to look at a landscape evaluation of the trapping protocol, again in this leading edge zone up here. And here we are looking at varying the number of baits per site, the configuration of the baits, like how to put them in a grid, and also the distance between bait sites. Or I guess this is number of baits, baited trees per site, actually. And this has been shown to be important, the number of baited trees in a grid, with because the whole purpose here is to have the greatest density of attacks on these baited trees, because then you can control more beetles that way. And the number of baited trees in a grid has been shown to be important in influencing the attack density on trees. And so that's why we're varying these number of trees in this grid situation. So our methodology, this is just kind of a snippet of our landscape where we will place our different treatments. Our treatments are, we have three main different treatments, which are 3 baited trees, 45 meters apart, which is the ESRD protocol. We're also going to do a grid of 4 and a grid of 8. And then if you zoom in, each one of these are going to be spaced at different distances from each other to try to make the most efficient and effective trapping protocol, and see if we can find if we can concentrate these and keep them from going to adjacent stands. So we'll have different distances between bait sites with a total of 45 plots that we're going to put out. And again, this will start in 2015 and be repeated in 2016. Yes. Okay, so when we come back to assess these trees, we'll put up the baits in the beginning of July. In the mid-September, after the flight is done, we'll come back to assess these trees in these baited areas. We'll take note of the number of trees not attacked, the number attacked, and importantly, the number of mass attacked trees. So that means we'll also look at the attack density of these trees on the north and south side. Again, trying to find a treatment that has the highest attack density on these baited trees. And we'll also do a survey around 50-meter radius of each of the baited trees to also look for attacked trees. On these trees, we'll also look at the presence of other bark beetles and try to come up with most cost-effective trapping protocol that has the maximum attack density on trees with the lowest number of baited sites put out into the landscape. And so if each one of these are baited sites, determining this optimum trap tree density and distance between baited sites, we'll be able to get at 4 main research questions. And the first one is, can these baited trees, baited trap trees protocol concentrate beetle attacks by attracting beetles to a defined area? And will this protocol be able to sufficiently detect small changes in population fluctuations of beetles while they're at these low densities? Will we be able to detect that with this? The third research objective essentially is that, can the baiting of selected sites provide substantial savings in search for infestations as compared to aerial surveys? And finally, can these tree baitings be used to prevent the dispersal from the baited sites while also allowing beetles to come into just these baited sites, thereby preventing adjacent stands from being infested? And with optimal trapping protocol, we'd hopefully be able to potentially reduce the beetle population through this direct control measure, also lead to savings in searches for infestation. And with this research, we'll also be able to look at some population information, such as relative population abundance, population fluctuations, as the beetle population in surrounding stands will be correlated to the beetle numbers that we catch in our baited sites. And also, these trapping protocols can also look at geographical distribution of mountain pine beetle and likely sources.